In this video, we're going to show you how to import a fastA file of your phage genome into DNA Master. So we will show you how to acquire the phage genome sequence from PhagesDB. We'll show you how to import that sequence, which is in a fastA file, into DNA Master. And then we will show you how to perform an auto-annotation of the genome, and finally save your annotated genome as a DNA master file, which has the extension of .dnam5. So I'm going to quit out of my presentation and open up PhagesDB so that we can acquire the fastA files of the genomes that you would like to look at. So if you wanted to look for QUK, you could type in QUK in the search bar, and it comes up as the first hit. And you can scroll around and look at all the new information that has been entered since it was sequenced. And here's a link where you can download the fastA file. But I would like to work with SheerCon today, and I wanted to warn you that Sally Special and SheerCon are really hard to find in this search bar, and I don't quite understand why. But there's another way you can acquire or find the phage page. You can go to the cluster list and click on that. It takes a second to load all the clusters. And then because I know SheerCon is a cluster E phage, I'm going to click on the E's, and then I'm going to go to the end of the list and then one page back to find SheerCon and click on it. And here we can see some additional information about the sequence of SheerCon. We know that ' genome length is 76,133, and we'll want to remember that. And we can see that it has a 3' overhang and the sequence of that overhang, the GC content. And here's the link where I can download the fastA file for SheerCon. Okay, now we don't need PhagesDB for a little bit. Go to your download folder, downloads, and copy this fastA file into a file somewhere on your hard drive, or for me, I keep it in Dropbox, somewhere where you can access that file later on. And I've already done that, so I'm going to click out of this, and I'm going to open up DNA Master. Now, if this is the first time you've opened up your DNA Master since you installed it, you want to go back in the annotation guide and look up how to update your DNA Master program, and also how to set the preferences of your DNA Master program. And there's a video posted on how to do that. So make sure that that's done first. And I usually click out of this Welcome to DNA Master box because I don't find it very helpful. What you want to do is go to the menu and click on File, Open, and then scroll down to fastA Multiple Sequence File. Click. Okay. This allows you to browse your folders and find the fastA file that you want to use in your analysis. And so I'm looking at, right now, the fastA file for SheerCon, so that's ready to go, and I click Open. And this will take a minute or two for the window to open up and show us that we've imported the sequence. This could take several minutes, and I will likely delete out some of this video so that you don't have to sit through all of it. In the meantime, I wanted to show you that I have the annotation guide open, and I have read through the annotation guide for this particular protocol. And that's because I easily forget the steps, and I would like you to do the same thing. Always have your annotation guide open while you're performing analyses in DNA Master. It shows you where to get your sequences, and it shows you how to import. We just did this step, and we're waiting for this window to appear. Okay, once this window has appeared, go down to the bottom right-hand corner of the window and click on Export, and select Create Sequence from this entry only. Okay, once the file opens for ShearCon, let's take a look at it before we perform the auto-annotation. This is the Overview tab, and it tells you basic information about your genome. Right now, it's only telling us that it's linear and has a length of 76,133. I want you to note the length of the sequence that you've imported into DNA Master, because we need to make sure that it's the same length reported on your phage page. And this is the correct length for ShearCon, so now we can proceed. The other thing I wanted to show you was the Features tab, and note that there are no features, and that's because we have not yet performed the analyses of the DNA, and therefore there are no predicted genes listed. And likewise in the documentation, there's really very little information other than the length of the genome and the overhangs. So let's perform the auto-annotation so that we can see where those genes lie. Go to Genome and scroll to Auto-Annotate. And you want to make sure that the settings for your auto-annotation are correct. And so I like to scroll down and make sure that my windows and selections in those windows match those in the auto-annotation guide. One thing I wanted to point out is that we're selecting both analyses, meaning we are choosing to have DNA Master run both Glimmer and GeneMark analyses. And in my annotation, I'm going to favor the Glimmer calls, which means the genes are going to be called based on Glimmer. But the program will also report to me if GeneMark has made a different call. What I would encourage people to do in their groups is have half the group click this button and half the group click this button and see how your annotations differ. It doesn't really matter either way because both calls will be reported in the notes section. Okay, you also want to make sure that both these boxes are clicked, insert template into comments, and examine and learn gene features. Now look at tags and comments, and you can see here all the template for the notes that I'm going to take for each gene are present. And you also want to make sure that the starting gene number is going to be 1 and that each successive gene is going to be +1. Sometimes it says 10 here, and you want these two numbers to be 1. Go to blast searches. Now some of you may choose to do the blast. However, this takes all night before all the genes are blasted, unless you're doing Sally Special, in which case it won't take as long because there aren't very many genes. So only click this box if you're able to leave your computer on all night so that the blasting can be performed in a long period of time. You don't want to shut down your computer while this blast is occurring. Because I don't have that much time, I'm not going to click that box. And then go to Automated Analyses, and we want to click on Select No Analyses. Now we're ready to click the Annotate button. And it says, Erase features and annotate genome. And yes, that's what we want to do. There's nothing in the features box. We want the analyses to occur, to be documented, and then parsed into the features. In order to do that, they have to erase what's already there. Okay, so the auto annotation is complete. This is a log of the analyses that were done, and we can click out of that now. And here we finally have our annotated genome, and in the Features tab, you can see that there are a lot of genes in SheerCon. In fact, they have predicted 139 genes. You can see that they're in different colors. The green ones are forward genes and the red ones are reverse genes. And I can click on these, and each time I click on these, I can get different information. We go back to the features. Those tRNAs are usually in blue. If we scroll down, we can see that they don't have a gene number because they're not genes, but here's where they're located. Here are their coordinates. And we have one for glycine, and we have another one for glycine. We have 2 glycines. Okay, so for now, I'm going to stop here and show you how to save your file. And you always want to go to File, Save as a DNA M5 file, and find a folder. You can save all your files right here in the archive within Wine, but I like to go to put things in my Dropbox. So I'm going to go to Sally, Dropbox, and store things in here. Genome sequences, SheerCon. And what I like to do is give it a really easy name. Since we have to keep the DNAM5 extension, I'm just going to call this SheerCon. And I'm going to give it a number. So every time I save something each day, I probably will give it a progressive number, because sometimes files get damaged and it's always nice to go back to the one from the previous day. And so you want to keep track of the progression of files that you have. So since this is the first one I'm saving, I'm going to give it the number 1 and push save. Okay, at this point you can click out of everything, and it's going to say save before closing, and you can say no because you've already saved it as a new file. And I would encourage you to save, every time you make changes in the file, is to save it as a new file each time you do so.